All right, let me tell you about the most mind-blowing instruction manual in the universe. No, not your iPhone user guide that you've never opened. I'm talking about DNA, the biological blueprint that's been crushing it for billions of years while we still can't figure out which way the USB goes in. Here's the thing. Your body contains about 3 billion letters of genetic code. If you printed that out, it would be a stack of paper reaching from here to the moon. And yet, somehow, it all fits inside something smaller than the period at the end of this sentence. Today, we're going to crack open this cosmic instruction manual and understand why it's simultaneously the most elegant and most ridiculously over-engineered system in existence. Trust me, by the end of this, you'll never look at yourself the same way again. Act 1. What even is DNA? The basics. Our story starts in 1869 with a Swiss dude named Friedrich Mischer who was doing what any reasonable person would do, messing around with pus-soaked bandages from a hospital. Delightful, right? He squeezed out white blood cells and found this weird, goopy substance he called nuclein. Mischer had just discovered DNA, but he had absolutely no idea what he'd found. It's like accidentally inventing the internet while trying to make a better toaster. Fast forward to 1953. This is when the real party starts. Two scientists, James Watson and Francis Crick, walk into a pub in Cambridge and announce they figured out the secret of life. And they weren't exaggerating. They discovered that DNA looks like a twisted ladder, or as we call it, the double helix. But here's what makes it genius. DNA is made of just 4 chemical letters, A, T, G, and C. That's it. 4 letters to write the entire instruction manual for building a human being. English has 26 letters, and we still can't agree on how to spell gray or gray. Meanwhile, nature built you with a 4-letter alphabet. Think of DNA like the world's most specific Lego instruction booklet. Each piece has to snap together in exactly the right way. A always pairs with T, and G always pairs with C. It's like a molecular dating app with a 100% success rate. No ghosting, no it's complicated, just pure chemical commitment. No ghosting, no it's complicated, just pure chemical commitment. Act 2, the architecture, how it's built. Let's talk construction. Imagine you're building the world's longest spiral staircase. The sides of the staircase are made of sugar and phosphate. That's your backbone, keeping everything stable. And the steps? Those are your base pairs, AT and GC, held together by hydrogen bonds. Now, here's where it gets beautifully weird. These 2 strands run in opposite directions. We call this anti-parallel, which is science speak for they're headed opposite ways but still totally work together. It's like a highway where traffic flows both directions on the same road, except there are never any accidents because chemistry is very, very good at following the rules. And remember how I said this all fits in your cells? That's because DNA is packaged like a masterclass in origami. It wraps around proteins called histones, forming little spools called nucleosomes. Then those spools coil around each other, and those coils coil again until you've got these super compressed structures called chromosomes. If you stretched out all the DNA in just one of your cells, it would be about 6 feet long. You have 37 trillion cells. Do the math, and your DNA could stretch to the sun and back over 100 times. And right now, it's all compacted inside you while you're probably watching this in your pajamas eating cereal. Nature is showing off at this point. Act 3, copying the manual, DNA replication. Okay, so you've got this incredible instruction manual, but here's the problem. When your cells divide, they need to make a perfect copy. And I mean perfect, like 1 typo per billion letters. For reference, if you tried to hand copy War and Peace, you'd make about 15,000 mistakes. DNA makes basically none. The process is called DNA replication, and it's bonkers. First, an enzyme called helicase comes in and unzips the double helix like it's opening the world's most important zipper. Each strand then becomes a template for building a new partner strand. But here's where it gets complicated and honestly kind of hilarious. DNA polymerase, the enzyme that builds new DNA, can only work in one direction. It's like a train that can only go forward, never backward. So this creates a problem because remember, the 2 strands are anti-parallel. So on one strand, called the leading strand, everything's smooth sailing. The polymerase just cruises along, making one continuous copy. Easy peasy. But on the other strand, the lagging strand, the polymerase has to work backwards relative to the direction the DNA is unzipping. So it makes these little fragments called Okazaki fragments, working in short bursts. And then another enzyme comes along and glues them all together. It's like if you had to write a sentence by starting in the middle, jumping to the beginning, then the end, then filling in the gaps. Inefficient? Absolutely. But it works because evolution is really good at making the best of awkward situations. It's basically the molecular equivalent of, if it looks stupid but it works, it ain't stupid. Act 4. Reading the manual. Gene expression. All right, so you've got your instruction manual. Now, how do you actually read it? This is where gene expression comes in, and it follows what scientists dramatically call the central dogma. DNA makes RNA, RNA makes protein. Information flows one way, very orderly, very dignified. Except, plot twist, it's not always one way. Retroviruses can go backwards from RNA to DNA. And there are these nightmare-fueled things called prions that are infectious proteins with no DNA or RNA at all. They're responsible for diseases like mad cow. Basically, they're the rogues of the molecular world, ignoring all the rules and causing chaos. But for normal, law-abiding genes, here's how it works. First, transcription. Your cell makes a temporary copy of a gene in the form of messenger RNA, or mRNA. Think of mRNA as a photocopy of one page from your massive instruction manual that you can carry to the workshop. This mRNA leaves the nucleus and goes to the ribosome, which is basically a molecular 3D printer. The ribosome reads the mRNA 3 letters at a time. Each 3-letter chunk is called a codon, and each codon says add this specific amino acid. Transfer RNAs, or tRNAs, are like delivery trucks bringing the right amino acids to the construction site. The ribosome links them together into a chain, and that chain folds up into a protein. And proteins do literally everything. They're enzymes, hormones, structural materials, transport vehicles. Proteins are the workers that actually run your body. Here's the kicker. You have about 20,000 genes, but your body makes over 100,000 different proteins. How? Through alternate splicing. Basically, your cells can remix the same gene in different ways to make different proteins. It's like having 20,000 songs, but creating 100,000 different remixes. Your genome is basically a very talented DJ. Act 5. Hacking the manual. CRISPR-Cas9. All right, so now we get to the part where humans looked at billions of years of evolution and said, that's cool, but what if we could edit it like a Google Doc? Enter CRISPR-Cas9. This is genuinely one of the most revolutionary tools in biology, and it came from studying how bacteria fight off viruses. Bacteria, those single-celled organisms we usually associate with spoiled food, have been doing genetic engineering for millions of years. We just figured out how to steal their technique. Here's how it works. You design a guide RNA that matches the exact DNA sequence you want to change. This guide RNA leads the Cas9 enzyme, which is basically molecular scissors, to the precise location in the genome. Cas9 cuts both strands of DNA, and then the cell's repair machinery kicks in. Now you've got 2 options. Option 1, let the cell repair it sloppily, which usually breaks the gene. This is called a knockout. Useful if you want to turn something off. Option 2, provide a template with the correct sequence, and the cell will copy that template. This is called a knock-in. Useful if you want to fix a mutation or add something new. This technology is already being used to treat real diseases. There are FDA-approved gene therapies for spinal muscular atrophy, certain inherited blindness conditions, and sickle cell disease. We're literally going into people's cells and fixing typos in their genetic code. But, and this is a massive but, we have to talk about the ethics. In 2018, a scientist in China used CRISPR to edit human embryos to make them resistant to HIV. Those babies were born, and their genetic changes are now permanent and heritable. The global scientific community collectively lost their minds because we don't know the long-term consequences of germline editing, changes that get passed to future generations. There's a huge difference between fixing a disease in one person's blood cells and editing the human germline. One affects only you, the other affects your children, your grandchildren, and potentially the entire human species. It's like the difference between painting your house and rewriting the city's building codes. Just when you thought you understood DNA, let me blow your mind one more time. Your DNA sequence is only part of the story. There's this whole other layer called epigenetics, chemical modifications that don't change the DNA letters but control whether genes are turned on or off. Think of your genome like a massive library. Epigenetics is the librarian deciding which books are on the accessible shelves and which are locked in the basement. The books haven't changed, but their availability has. Here's where it gets wild. These epigenetic changes can be influenced by your environment, your diet, your stress levels, and sometimes they can be passed to your children. That's right, your lifestyle choices might affect not just you, but potentially your kids' gene expression. For example, studies have shown that children and grandchildren of people who experienced famine may have altered metabolism. The trauma of starvation left an epigenetic mark that influenced the next generation. It's like your DNA has a memory, which is both fascinating and slightly terrifying. And then there's the fact that 98% of your genome doesn't code for proteins. For years, scientists called it junk DNA, which in retrospect was incredibly arrogant. It's like buying a computer and calling everything that isn't the keyboard junk. Turns out most of that non-coding DNA is regulatory. It controls when, where, and how much the protein-coding genes are expressed. Your genome isn't just an instruction manual, it's an instruction manual with footnotes, appendices, commentary, and a very opinionated editor making real-time changes based on what's happening in your life. Conclusion, the big picture. So here we are, DNA, a 4-letter code written 3 billion letters long, copied with near-perfect accuracy every time your cells divide, read out in an incredibly complex 2-step process, regulated by an entire secondary layer of chemical modifications, and now editable by humans with CRISPR technology that costs less than a used car. Every single cell in your body contains this complete instruction manual, and it's been refined over 4 billion years of evolution. It survived ice ages, asteroid impacts, and that one time you ate gas station sushi. It's the most robust data storage system ever invented, and it runs on chemistry. But here's what really gets me. We're the first species in Earth's history that can read and edit our own source code. That's simultaneously amazing and terrifying. We have the power to eliminate genetic diseases, but we also have to decide where to draw the ethical lines. The future is genuinely wild. We're talking about designer babies, bringing back extinct species, curing cancer, extending lifespan, all on the table. But with great power comes great responsibility. And unlike Spider-Man, we don't get to just web sling away from the hard ethical questions. Your DNA is a masterpiece written by evolution, edited by your environment, and now potentially revised by human choice. It's the ultimate instruction manual, and unlike your IKEA furniture directions, there are no leftover pieces. Thanks for watching. If your brain hurts, that's normal. We just covered 4 billion years of evolution in 14 minutes. Hit subscribe if you want more science explained with inappropriate analogies. And remember, you're more than your DNA, but your DNA is pretty incredible.